optimization. So the number of runs that are possible to run in a given DOE or optimization campaign is a primary constraint when we're thinking about how to design an optimization strategy. So this is particularly true when the final process we're optimizing will be run at large scale, and where the runs of actually running that large scale can be prohibitively expensive. So this sort of situation is very common in bioprocess development, where it's a common strategy then to use scaled down systems to run our optimization. And in this talk, we're going to hear about some of those scaled down systems that can be used and how they can be used to do effective DOEs to get a good knowledge of the process landscape and to establish a process that we can then scale up ready for production. So please do ask questions as we go along in the question box in the webinar, and we'll be addressing these towards the end of the talk. So now I'm excited to introduce our presenter for today's webinar, Dr. Lucia Dunajová, who's a senior research scientist here at Synthace in the lab team here. So Lucy works at the intersection of science and engineering to push forward the Synthace Life Sciences experiment platform. Her recent work has focused particularly on using Synthace and high throughput process development tools to exemplify protein purification applications. Prior to joining Synthace, Lucy was a senior scientist at Oxgene, where she engineered cell lines for lentiviral production, and she has a PhD from the University of Nottingham. Thank you very much, Lucy, and without further ado, I'll hand over to you.
Thank you for the introduction, Marcus. And so I would like to take the opportunity to get a poll out to you, just so that we get a bit of a feel for how much you use DOE in your organizations. So you should have a poll coming up just about now, where hopefully you can tell us how often do you guys use DOE in your day-to-day experimentation. So I'm quite curious to see what's the spread out there in terms of its usage. And we're just going to give it a few more seconds, just so that everyone can get their answer in. And these are the results. Oh, here they are. So wow, it's great that there's a portion of our attendees who are actually using it quite regularly. And it seems like there's still a small bunch that needs to be convinced. So yeah, that's definitely great to hear. I would be particularly interested in seeing what the 2% who have used it, but it hasn't worked for them, what's happened there. But cool, thank you so much. And without further ado, let's get started. 1 minute. Okay, here we go. Sorry, you get to see the amount of screenshots I get to take. Sorry about that. So yeah, let's dive right into it. Thank you, Marcus, again for the great introduction. So today I will be talking to you about, give you a brief recap of what is actually DOE methodology. Then we're going to dive a bit into what tools there are available for employing DOE for purification optimization. And then I'll walk you through some examples of where we specifically apply DOE in purification, and finally closing with some brief summaries and then going into the Q&A session. So what is the design of experiments and why should we use it? Here I would like to take the opportunity to highlight a few really useful resources, some of which we have mentioned in one of our previous webinars. And so namely this paper, Lost in Space, Design of Experiments and Scientific Exploration in a Hogarth Universe. It's really useful because it nicely explains why you'd want to use DOE, and I will actually show you a few key slides from few key figures from this paper on the next few slides to help me make my DOE case really. Next up, we have this a bit more in-depth book called DOE Simplified, which also gives some nice practical suggestions for getting started with DOE. Next up, we have actually generated our very own guide to design of experiments aimed specifically at biologists. And finally, there's this really nice resource freely available online, which is specifically tailored to using DOE in protein production and purification from GE Healthcare and now Cytiva. So I definitely urge you to check all of these out. So in biology, the traditional way of designing experiments is generally to change one factor at a time while fixing all the other variables and measuring an effect of this factor on a particular response. That's why we tend to call it one factor at a time approach, or OFAT, as the catchy name as it is. And so imagine a scenario where you're looking at a particular factor, let's say a temperature, which you're increasing to try and see if you can improve your protein production or your protein expression. And what you find that indeed with increasing temperature, you get better protein yields. But so imagine a following scenario where your collaborator, for example, does the exact same thing. They increase the temperature, but find that the converse is true. Their protein yields actually decline with decreasing temperature. So what has happened here? How is this possible? Well, for starters, both groups have actually valid data. But what has happened is that there's an undiscovered interaction between the factor both groups were investigating, factor 1, and another factor, factor 2. And the difference here lies in the fact that group B has set this factor level to low, whereas group A has set this factor to high. And so kind of by chance, both of these groups have stumbled on a process setting which is actually which has the desirable outcome of increasing protein production. But as you can hopefully appreciate, both of them lack a more thorough understanding of their experimental space. And so that's a lot of information that they have missed. And this is in contrast to DOE, which actually follows a statistical framework where factors are systematically varied in parallel. Like you see here, you vary both factor 1 and factor 2. And so what this means in practical terms is that you actually end up with a smaller number of experiments, as you can see here with the number of dots. But at the end of it, you actually gain a better understanding of what factors and how they may be influencing your system, or in this case, your purification process or protein production process. And so it goes without saying that having this kind of understanding is key to being able to not only control your process, but also predict any potential outcomes on your key responses if you changed any of the process settings. And so here I'm just showing pretty much the same thing, but which really nicely highlights the level of effort you really need to put in with this OFAT approach, where you're really doing more and more experiments, but at the end of it, you're still really none the wiser. Whereas with DOE, with one well-designed experiment, you can already gain so much more knowledge. And so, however, there's many flavors or types of DOE design, which are based on your ultimate objective. And despite of how powerful DOE is, it doesn't necessarily mean you just run one experiment and be done with it. It's still a very iterative approach, similar to the OFAT approach in this sense, at least. So what might happen at the beginning, you might start here on the left, where you're just trying to understand which factors are actually important, what has an effect on your system. And so this would be quite a wide design, looking at a large number of factors, and you're slowly moving more towards the right-hand side, where you're narrowing down the number of these factors and honing in on a very concrete factor range. And finally, you end up somewhere here on the right, where you're looking at understanding whether your process is robust at the factor levels that you've chosen. And so each of these DOE types can be broken down into 5 fundamental steps, which in many ways is similar to how you would traditionally approach designing a biological experiment. So the first step is really the most critical step, because it's here that you establish what is actually the question you're asking, or what is the goal you're trying to achieve. To this end, you gather all the available knowledge, maybe even run some test experiments, and you use this to define your factors of interest, your factor levels. But also it's important to start thinking about what are your responses and how are you going to be measuring these. And especially understanding what is the level of error or noise in your measurement systems, as these can then impact the final analysis. Next up, then you have to start thinking about, okay, if I want to run this particular experiment, how much resource do I have to do it? Because this is what determines what type of design you're actually going to be able to run. And so generally we use a statistical software package to give you this design, considering practicalities such as budget, time, equipment. And then once you get this design, you go in the lab, you run it, you collect all the data that you wanted to collect, and you structure it in such a way so that it can be fed back into the statistical package. And finally, you build models, which is a fancy way of saying you look at the cause and effect relationships between the factors and the responses, and using some of your domain knowledge, you start to draw conclusions about your process. And so we've previously really tried to bring home the message that DOE doesn't differentiate. You know, you can really apply it to anything across the entire bioprocess pipeline. And so previously, we have shown specific examples pertaining to the cell line development area and the assay development area. But today, like you know, we're going to be focused on downstream purification. So in downstream development, there's a number of key high throughput development tools available, shown here on the left-hand side. And what's great about them is that they are highly amenable to the use of DOE, given the fact that they provide this option of testing many different factor level combinations at the same time for a fraction of the cost of these higher scale DAP models over here that you see here. Therefore, what this means is they are a great starting point for gathering relevant information about factors, so which factors actually matter, and the factor levels before moving towards these more expensive scale-up runs. As here, with the increasing column sizes, each run becomes dramatically more expensive, not just in terms of cost of the materials, but also of time. Until hopefully we can arrive at a very robust process that we can transfer to production scale. So let's take an overview of what tools are there. So from the picture I showed you, we have the pre-filled 96-well filter plates, where one of the main suppliers, I believe, is Cytiva, so-called predictor plates. Then we have the pre-packed micropipette chromatography tips, phi tips. These are a bit strange considering they have dual flow, so they're perhaps less of a common purification tool used for this purpose. And then we have miniature pre-packed columns, where the frequent supplier is actually Repligen with Opus robocolumns, where you can get them pre-filled with resins from across many different suppliers. And then we have the predictor robocolumns from Cytiva. So the main advantages of all of these tools is that, obviously, because they're so scaled down, they use a much smaller sample volume and a small resin volume, which in turn translates to lower material costs. Or perhaps keeping the cost same, but what it enables us to do is to run a larger number of parallel experiments, which in turn translates to much shorter development times. So imagine one benchtop scale run is still one column, whereas with this, you can run many in parallel. And what's also important is that they're automatable, which means you don't have to do this manually. You set up the method and walk away. And most importantly, there's many studies out there that actually show there's really good correlation with lab scale columns, both in terms of yield and also various other product characteristics. However, it's not all fine and dandy. There's, of course, some limitations to this, and that is mainly that you need to run your entire purification process before you actually get any data out of it. So you collect your fractions and then perform offline analysis of these for example, absorbance at 8-80 to actually get any data. And unfortunately, because they're using automation, you might require significant automation expertise to actually program the methods needed to run them. But luckily, there are tools out there available which can really simplify this process. Wink, wink. But unfortunately, even if you really narrow down the factors required and the factor levels, you still require validation and optimization at larger scale. But of course, the amount of cost here will be much, much reduced because you won't have to run the same number of runs. Next up, we have specifically 96 well filter plates, which you can see here in the top right corner. So as the name suggests, they come in the format of a 96 well plate, which means you can actually run 96 unique factor combinations at the same time, so in parallel. And they come pre-filled with either low or high volume of chromatography resin. And once again, you're really spoiled for choice when it comes to the types of resins available. Here, I actually have to highlight that it's great that these plates can be used both manually and also on automated liquid handlers. So manually with a centrifuge or a vacuum manifold. So you can imagine a scenario where you combine both. So you do parts manually, part on the liquid handler. And the applications most commonly for these used with these types of tools are for initial screening, or perhaps you want to really thoroughly investigate a defined space in your system. And otherwise, they've been really used for all of the stages of the purification process. So this is your binding, wash, elution, cleaning in place, resin screen, and even adsorption isotherm studies. Because the interactions between the resin and the sample pretty much remain the same as in the column. And so here, the type of study you decide to do really determines which filter plates you actually need to purchase. As for binding studies, you're effectively just looking for overloading the resin, so you can get away with really low resin volumes. Whereas for wash and elution studies, since you're trying to characterize the level of impurities, you're kind of limited by that detection limit of those impurities. You need to go for high volume resin generally. And so finally, we have robocolumns, which I'm, to be honest, the most familiar with. And you can see them here. They have this cylindrical shape, and into which we inject our sample using these stainless steel tips on the Tecan platform. So whereas the other tools are actually liquid handler agnostic, it doesn't really matter which one you use. This tool specifically can only be used on the Tecans. And so they're actually almost the most representative scaled down model of an actual column, because as you can see, it has that shape. And so what happens is, because you're injecting the samples using an 8-channel head, you can effectively only run 8 experiments in parallel, and then you can move on to the next 8, wait for them to be finished until you do the next 8. So this slightly lowers the amounts of experiments run in parallel. And they're likewise available pre-packed with various different resins and at different volumes. So you can decide based on how much sample you have available. And most importantly, they have a linear flow rate controlled by the dispensing speed of the liquid handler, which means you can get actually equivalent residence times and equivalent protein loading capacities as you would in a lab scale column. The main limitation of them, though, is that you get intermittent flow, subject to the fact that the liquid handler sort of goes away, takes some sample, injects it, and has to go back to take some more. And that is what happens there. And so the applications here also are for screening and for optimization of chromatographic conditions. But generally, you tend to employ robocolumns once you are in a slightly narrower space. As you can see, the number of runs possible in parallel is a bit lower. And finally, another application which is perhaps a bit less relevant for this webinar, you can also use this technique in conjunction when you're actually doing maybe DOE in your upstream process development, where you're just trying to generate enough product so that you can run your quality assessments. And finally, down here, I'm showing a pseudo-chromatogram generated from the data absorbance measurements collected from these collection plates. So you get data that looks very similar to your standard unicorn platform. So it's really, really great. And so how do you choose which tool is best for your purification goals? So first of all, you really just need to think about what is the actual objective of the experiment. Are you trying to screen out which factors are relevant, or are you already trying to set what are the optimum settings of your important factors? Or finally, do you have an already established process and you're just trying to understand whether it's robust? And finally, what you of course have to think about is, again, what is the availability of your equipment, your budget, your sample, and then of course your time. How much can you dedicate to such an experimental campaign? So next up, I'm going to dive a bit more into specific examples in how to use DOE in purification. So for one, I will be showing data, but for another, it will be more of a theoretical walkthrough. So first up, we were trying to basically characterize purification of E. coli express transaminase. So transaminases are enzymes that are becoming increasingly important in the chemical industries. But to us, they were pretty much unknown. We had very little prior knowledge of the purification process. All we knew is that we wanted to use anion exchange gradient elution chromatography from clarified E. coli cell acid. So the focus was really early stage exploration with the aim to maximize yield and purity, as it always is, isn't it? And so here I'm showing the type of tool we decided to go with. Since we're trying to do gradient elution, the tool of choice is robocolumns, as these can actually mimic actual gradients. And so here I'm showing a sort of typical purification process. As you can appreciate, there's several stages, each with loads of parameters that you could potentially investigate, which may or may not influence each other across the stages. We decided to focus on investigating the resin type and then various parameters across the load, wash, and the gradient elute stage. And so since as I've already said, we really had no prior knowledge, we decided to go with a type of design called space-filling DOE design. And here we chose to investigate 48 unique combinations, factor combinations or runs, which resulted in 48 separate robocolumns. And so you can see already here in this diagram of the design is that this type of design really isn't your usual one that I've shown before. It rather prioritizes coverage within the design space. And so if you're not as familiar with this type of design, I will briefly cover what are the differences between the traditional DOE and the space-filled DOE. So again, here I'm showing a diagram of the traditional DOE flavor kind, which is really quite orthogonal. So by that I mean each factor level is replicated multiple times within the design. So for example, for factor 1, you have this +1 setting. It's actually repeated 4 times in this specific design example. And that is because it's aimed at reducing aliasing of the different effects. So in other words, it's aimed at being able to correctly distinguish which factors are actually impacting the response. But the limitation of this type of design is that you do need to have some prior knowledge. You do have to make some assumption about your design space boundaries, meaning you wouldn't want to choose factor levels which give you 0 response, because then your DOE modeling will effectively fail. And so what happens is you might end up in a situation like this, where these 3 points are your responses that you've measured. And you can see here that if you do generate a DOE model based on these points, you end up with something like this, where you've effectively missed this local maxima. What this means is you may need to do multiple iterations of DOE to actually understand this full factor profile. In contrast, in space-filling DOE, we are sampling the space uniformly. So that means we don't actually need to know that much about our system, because we're pretty much going to try everything, right? There's no just 2 levels that we're investigating many. And so what this means, you end up with something like this. You actually cover the experimental space pretty thoroughly, and you can easily identify your factor profile. And likewise, you identify dead spaces, so meaning where your response is 0 and where you probably shouldn't spend much more of your time. So let's take a brief look at the overall results of this space-filled DOE. So here, each of the rows is the different resin type over here. And the bars in the plot represent the yield of the individual collected fractions in this robocolumn purification run. And the horizontal line over here indicates 80% purity. Shade depth of each color represents yield of best pool, so pulling adjacent fractions together, which fulfill this 80% cutoff, 80% purity cutoff. And then the runs are organized from best to worst, really. And so you can see clearly that for some conditions, we really didn't hit the jackpot. They didn't perform so well. But likewise, we were able to identify conditions which really weren't so bad. And so designing further DOEs, this would be perhaps a good place to start. And so next, we used our results where we tried to use a slightly less stringent response to what I just showed, and used a cumulative pooled yield instead without being so strict about purity. And so we built a quadratic model. And so here you can see the significant factors and their profiles over here. At the optimal process settings, these factors in red should be set to low. So unsurprisingly, your sample mass loaded being low, your flow rates being low, and your pH should be set to high. So for wash and elute stage should be set to high. And at this point, you start to get hints of interactions across the different stages, which really speaks to the importance of not just limiting yourself to one particular stage, but really covering the entire purification process. And it's quite interesting to observe that you get this inverse interaction between, for example, flow rate and the elute pH, where perhaps unsurprisingly, load flow rate is generally better for yields. But imagine this is a bottleneck and you want to speed up your process so you have a shorter execution time. You might want to consider lowering your elute pH so that you can still compensate for this increase in the load flow rate. And so next up, I want to continue on and return to the area of traditional DOEs. And at this point, I would want to look at how we can maximize host cell protein clearance in monoclonal antibody purification. So like I said, this will be more of a theoretical example where I will not be showing any data, but rather focus on the design phase of the DOE. So for those of you who are maybe not as familiar with this, I will just give a brief overview. So monoclonal antibodies are typically expressed in a host cell organism, such as Chinese hamster ovary cells, and then they are this monoclonal antibody is then secreted into the cell culture media. And so we see that here, the cell culture media is applied onto the protein A column, which specifically interacts with the antibody. Then we wash the column using a neutral pH buffer, and finally elute with an anesthetic solution. However, the final product, despite the fact that the yield of this capture step is already pretty high, we are still getting some host cell protein contaminants coming through from this culture medium. And of course, this is very undesirable, as the final product, the monoclonal antibody, is intended for a patient. So we want to minimize any amount of impurity so that we prevent any detrimental side effects in the patient. And so what many people have started doing is to include an additional wash stage specifically aimed at reducing the level of these impurities in the final product. And so I've taken a look at the type of studies that are out there, and effectively what a lot of them have done is taken this typical OFAT approach where they've simply chosen a wash buffer at a particular pH and then experimented with including various additives with the aim of disrupting non-specific interactions between the host cell proteins and the bound antibody. And so you see in this example here, let's say they have chosen a wash buffer at pH 5. They would either add salt or maybe detergents or maybe both, and then include a control condition perhaps at a neutral pH, which in my example was PBS, for example. See what results that gives, and then move on and do a second experiment where they would effectively repeat the same thing, but maybe move to a wash buffer with a different pH. And going back to what the slides I've shown you previously, this once again amounts to increasing time and cost in terms of the amount of experiments you have to do. And so once again, with a traditional DOE, you cover this experimental space pretty easily, pretty completely, and you get much better understanding. And so what I've set out to do is design a DOE that would systematically explore the very same factors reported in literature. And the goal here is to look at the effect of wash buffer pH specifically alone or in combination with any of these additives, which are quite different. So they are expected to disrupt the interactions in different ways. And you might notice here, I've included these two groups where I've added either urea or guanidinium hydrochloride, and another group, detergents, where we have tween 80 or chaps. And so at this point, we have to also think about our responses, which of course would be lowering the whole cell protein levels. But likewise, since these additives act by disrupting non-specific interactions, we have to be careful that they're actually not disrupting the interaction between the protein and the resin itself. So between the monoclonal antibody and the resin itself. So we also have to ensure that one of our responses is recovery as well. And finally, since these additives can once again have some impact on the protein, we need to make sure our quality is also maintained. So at this stage, we may already decide to include some particular quality metric. And so going back to the groupings, I would like to just highlight this once more. You might have noticed that I've assigned two different additives to a group called denaturients and another two additives to the group of detergents. And so the reason why I've done that is that because it probably doesn't make sense to include both of these in the wash buffer at the same time, since their mode of action is likely very similar. So I've used this method to effectively specify constraints, which you can normally do using a specific tick box in the statistical software. But in this way, we are actually not limiting our design space at all. And what's more, this specifying constraints in this manner is actually very scalable to including additional types in each group. So you could imagine adding more additives here if you so wished. But of course, here you're making the assumption that these reagents will act in a very similar way and will produce a very similar response. So here I'm showing you the interface of the JUMP statistical software. We have simply added in all our factors and specified what type of factor they are and what levels they're at. Next, we want to begin defining what are the terms of interest to us. So what are we actually looking to get out of this experiment? In this instance, I've defined the main effects only. So we're looking at simply which factors have an effect on our response, if any. And you can see the statistical software already gives back the recommended amount of runs that it needs to get to be able to provide you with a good enough model. And so at this point, it's you should start thinking about your resources, as mentioned previously. And so you see, at this point, you might think, right, do I want to execute this minimal design using 2 sets of 8 robocolumns? Or perhaps expand my design further to include 2-factor interactions? And as you can see, at this point, this gives you 52 runs as a recommended number. So filter plates with 96 runs available seems like a more feasible choice. But this still leaves you some space on the plate. So why not add more terms to our model? And so in this instance, I've decided to expand the design to include third-order interactions, but with pH only. And the reason I've done this is because using some domain knowledge, I'm estimating that the pH, through its effect on protein structure and stability, will likely play the most important role in the non-specific interactions between the host cell proteins and the antibody, as well as for maintaining binding to the resin. But so here we are with our 88 runs for our DOE design, which means we still have 8 runs available on our filter plate. So though we could easily specify manually this number to 96 and actually expand our DOE design, it's important to not forget good experimental practice. And what I mean by this, it's still really important to include known performance control conditions where you know what the response is going to be. And what's more, and you can actually manually add these conditions to your DOE design and then use it later for modeling. Additionally, having these types of controls is actually really essential for when you're performing DOE iterations, as then you have a consistent benchmark between the different experiments, and you can easily distinguish whether any odd responses you may see is due to DOE implementation or whether one of the factors is off, let's say your reagent. So I really recommend adding those always in. And so here you have our final DOE design table, where, like I said, down here I've included manually some control conditions where we're simply using a simple neutral pH buffer with no additives in. But you might ask yourself, wow, well, you didn't actually include that many factors, and you would be right. There's additional factors which I have fixed in this theoretical DOE example, which are likely to have an impact on the system, on the process. And specifically, since I've mentioned that it's very likely the host cell proteins are interacting with the monoclonal antibody itself, you can imagine how the antibody type, so that the antibody that you're actually trying to purify, will have quite a significant effect in this scenario. And what might happen is that the factors that you identify in the DOE might differ in their importance for the different antibody types. And so the solution here is simple. You simply need to replicate this design by using the different antibody types to perhaps start building a more generalizable process rather than something that's very tailored to a particular molecule. And so finally, I want to leave you with this, with what I think is likely going to be the future in bioprocess development pipelines. And it's this typical diagram where we sort of separate out these different areas into these different buckets. But we know from the many studies that upstream conditions, for example, have a very significant impact on the downstream purification processes. So it sort of ends up being a balancing act between the different areas. And going back to the previous example of host cell proteins, we have actually seen this in-house as well, where we have performed an AMBER 250 study in CHO cells producing a monoclonal antibody, where we varied pH, temperature, and agitation, and then simply taken these bioreactor cell culture supernatants and purified them using the same protocol using robocolumns. And here in this small plot, you can see that though the differences between the different bioreactors wasn't particularly huge, there are definitely variations. And you have to remember, we didn't look at such things as media composition, and you can imagine that having a really significant impact. So I would like to encourage you to maybe think about applying DOE in a more holistic way, where you consider all of the different factors from across the different areas into a single DOE design to perhaps, make sure that you have a bit of a more overview approach to your process. And so finally, I would like to acknowledge the people that did a lot of this work, so namely Emily Fritsch and Jordan Ang, who are unfortunately no longer with us. At Synthace, I mean, they're very much alive. And these guys have done basically all of the space field DOE work. And then a big shout out to Sid, who's helped me a lot with pulling the DOE example together for host cell protein. And then finally for Marcus for being the guiding star of helping me pulling everything together at the end. So that's it from me. Thank you.